While we were scrambling to control COVID-19, amphibians have been suffering from their own pandemic for years now. Since the mid-1900s, a flesh-eating fungus has been condemning more species to extinction than any other pathogen ever recorded.
Chytridiomycosis in amphibians is caused by two species of fungi, Bacterium dendrobatitis and Bacterium salamandrivorans, visibly affecting the skin and eventually killing them.
Fungi have also been wiping out millions of other animals, such as bats. Besides being major components of many food webs, these animals consume insects equating to hundreds of billions of dollars worth of pesticides and help reduce the spread of disease, like malaria. The COVID-19 pandemic has taught us how important diagnostics is in curbing the spread of disease. Similar to COVID-19, the fungus spreads with travel in the form of trade. However, testing at ports has been impossible since results are quite latent and expensive. In the wild, after sampling, analysis is done by a trained professional using PCR in lab, but getting the result takes ages. Once confirmed, finding the patient again is impossible. The NYU Abu Dhabi iGEM group is on a quest for a solution. We are developing a rapid point-of-care diagnostic device for fungal disease detection. In November of 2019, after the NYUAD iGEM group returned from the Giant Jamboree in Boston, a conservation research scientist from the University of Friedberg reached out to the team and proposed a problem for the group to address.
Dr. Penner explained the issues he currently faces with diagnostic methods, and he indicated that a rapid diagnostic device, much like our past solutions, would be a game changer in monitoring amphibian disease populations. We found that interesting, so the first thing we did is try to understand amphibian decline, the related fungal disease, and overall biosecurity when it comes to animal infectious disease control. So now that we had a fairly good grasp of the problem, we set out to develop this point-of-care, rapid fungal diagnostic device. To do so, we had to imagine a pathway, and this is where the past experience with NYUAD iGEM project comes in handy. A pathway that begins with the first user interaction. And this gave us a master plan that we could work with.
As a precursor to defining the pathway, however, we had to select our detection method.
We needed to determine the optimal pathway to take in order to see if there's detection for BD and B-cell fungal infection in the amphibian species. This first relied on us determining a marker for what we would use to identify that the fungi is present. From nucleic acid detection to toxins, chemicals, and other antibodies, we ultimately settled on nucleic acid detection. Our literature review pointed us in the direction of DNA over RNA because it would be easier to work with. So finally, we had to decide on a specific region of the BD and B-cell DNA for which we wanted to detect. Our focus was on finding something that was highly conserved and non-pathogenic, so it would be safe and accurate to use. We decided upon the ITS region, the internal transcribed spacer, which is a highly conserved spacer DNA, which does not encode for anything, meaning it is extremely safe to use. It is also the region that is used as a barcode region in fungi detection. After consulting with some experts in the field, we got confirmation that our choices for both nucleic acid detection and the use of the ITS region as our target were appropriate for fungal detection.
The first stage in the diagnostic pathway is collecting the sample.
The standard method for sample collection is to swab the skin of the amphibian. Recently though, eDNA, which is sampling the habitats of the amphibians itself, for example ponds, lakes, etc., has proven to be effective, and our team is planning to have solutions for that down the road. However, the focus of the current project is still on swabbing.
After gathering this mix of genetic material, the next step in the diagnostic pathway is to extract and purify the DNA.
While in a lab, this can be done using a centrifuge. In a POC setting, this is a major challenge. We started out by mapping all the options that we could have in a POC device, which you can see here. And then based on our constraints and requirements, we narrowed these options down to what you see right now on the map. So these options are bead beating, which is mechanically lysing the fungal spores using rapidly moving beads. It's a recently described combination of bead beating, sonication, which is lysis using acoustic waves, and atomization, which is another form of mechanical lysis by passing the spores through small orifices. The other options are silica matrices with magnetic extraction, where the lysate is purified. Basically, the detergent is removed so that it doesn't hinder the downstream reactions by using magnetic beads that selectively bind to the DNA. Another option is lysis buffers plus silica matrices, which also selectively bind to the DNA and help in purification. And then there's Whatman FTA paper, which is a paper that's enriched with lysing capabilities and can lyse with only a few washes. So we're designing experiments to test all these options.
We then use ultra-sensitive and high-throughput nucleic acid detection methods by coupling an amplification reaction like RPA and LAMP with a targeting enzyme like CRISPR-Cas12a or CRISPR-Cas13a.
Keeping in mind the end result usability of our product as a field detection system, we identified 3 key design considerations to take into account for our amplification reactions. We wanted it to be isothermal, meaning the reaction would happen at one constant temperature. We wanted it to be rapid detection and have high sensitivity. This is so that field users would not have to carry around clunky, expensive equipment, and they would get fast results that were highly accurate. So from this, 2 amplification techniques emerged: recombinase polymerase amplification, or RPA, and loop-mediated isothermal amplification, also known as LAMP. And we wanted to test the difference between these 2. So we decided to design our RPA and LAMP primers, which we also added as parts to the iGEM registry, against our target ITS region. So here's a little bit about how RPA works. So here we have our RPA primers, which are designed for the fungi ITS DNA region, and it will attach to this region if it is present in our collected sample. From there, we have these recombinase and polymerase enzymes, along with other reagents, that will work quickly to clone these sequences, to amplify and make more of them. Specifically speaking, recombinase will help insert the RPA primers into the DNA, which will then begin to separate into 2 single-stranded sequences. And from there, DNA polymerase will help extend this DNA. Eventually, you'll end up with 2 identical DNA strands from 1. And this reaction repeats itself, and exponential amplification will occur from just a small amount of starting DNA. However, because RPA occurs at a relatively low temperature, the separated single strands of the DNA can easily rebind together, which is unfavorable because it would interfere with the DNA replication process. So, to make sure this doesn't happen, a family of proteins called the single-strand binding proteins, or SSB proteins, will bind to the separate strands to prevent them from rebinding. Due to limited time in the lab, we were only able to test for RPA and not RPA and LAMP. The primers we designed worked with their respective targets. So the BD primers helped to amplify the BD DNA, and the B-cell primers helped to amplify the B-cell DNA. We confirmed amplification by visualizing using gel electrophoresis. When we ran the gel, we saw a band at the length corresponding to our amplicon region, showing that the RPA primers were functional. The target sequence in the sample matched the corresponding primer set. When we tried to amplify the BD DNA with the B-cell primers, we did not get amplification. Conversely, when we tried to amplify the B-cell DNA with the BD primers, we did get amplification. Now this shows that our RPA primers are specific to their respective BD or B-cell DNA targets.
In order to increase specificity, our team decided to add a second layer of detection by targeting our amplified RPA product with the CRISPR-LB-Cas12a complex, which is derived from the Lactospiraceae bacterium. This combination is called detector, which is otherwise known as a DNA endonuclease targeted CRISPR transporter. Ultimately, this reaction coupling achieves attomolar sensitivity.
Now, what exactly is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it allows researchers to easily alter DNA and modify gene function. The CRISPR system that we are interested in is the Cas12a system, which is one type of the genome editing tool that we found promising. The Cas12a protein is a targeting enzyme that binds to and cleaves DNA. The guide RNA is a complementary sequence to the target DNA. The guide RNA helps the Cas12a enzyme to recognize the target sequence, which will then activate the Cas12a enzyme. Now, to incorporate the CRISPR-Cas12a complex into our detection pathway, we had to design our guide RNA to be complementary to a location within our amplified region, so within the ITS region of BD and B cell. Now, the activated Cas12a enzyme changes its shape slightly, and this new shape allows a DNA reporter molecule to bind to and be cleaved or cut by the enzyme. Now, this cleavage activity indicates the presence of our targeted nucleic acid sequence. Because remember, if our target region, our ITS region, is not there, our Cas12a enzyme would not be activated. Its shape wouldn't change slightly, and it wouldn't be able to cleave the reporter molecule.
So in the lab, we were able to experiment with the detector protocol, which is both CRISPR and RPA, on our target gene. So, starting with a triplicate of our prepared samples containing our BDG block, running our optimized detector protocol yielded positive results. Based on these results, we have a working amplification and detection pathway. We have every reason to believe that our detector protocol will also work with our B-cell ITS region and its corresponding primers and guide RNA. So, for our future plans, we plan on investigating the best combination of amplification and detection techniques in order to identify the most efficient detection pipeline. And to do that, we plan on determining the detection limit of each combination.
Going forward with our project, we plan to implement a positive control in our final multiplexed POC device. This is to ensure that our device pathway is fully functional. A promising gene to be used as a positive control is the mitochondrial DNA 16S ribosomal RNA gene, which is a well-conserved sequence that is universal almost across all amphibians. In addition, as we are currently experimenting on E. coli cells, we plan to extend our detection approach to actual chytrid fungi.
And all these reactions are happening in what we call the reaction medium.
And we looked at a wide range of case studies to do that, so that we could see what kind of device platforms had been described in the literature. And these options ranged from microfluidic chips, paper-based biosensors, droplet cartridges, to even completely integrated solutions like lab on a chip. Based on these options, we're brainstorming and adding our own additions, which you can see mapped here. And for example, these are a droplet cartridge with pH changing capabilities so that DNA can be transferred throughout the device just using magnetic beads, or having robotic automation of manual pipetting, or using PCR tubes that have pierceable membranes so that fluid can be transferred throughout different stages vertically, or having magnetically operated systems that have small gates or piercing rings. And we're using all these options and combining them with other device processes to create pathways that will be tested soon.
Upon CRISPR-Cas cleavage of our reporter molecule, a signal can be released in two different ways, either through fluorescence or through a lateral flow assay.
So, in order to detect the activation of our CRISPR enzyme, our first option is to use a quench fluorescent single-stranded DNA reporter. When our CRISPR-Cas system is activated, this reporter is cleaved, giving off a fluorescent signal that can be detected. Meanwhile, our lateral flow immunoassay can also be used to detect the activation of our CRISPR-Cas system. Now, this assay will take place in a strip with a test and control line. And in essence, the absence of a test line is a negative result, while the presence of a test line is a positive result. Now, this detection is achieved by using a dual-labeled fam-biotin single-stranded DNA reporter molecule. And so when our reaction mixture is applied to the strip, the fluid will be able to flow through the strip by capillary action, passing through the test and control line. Now, the first line is a control. Through the binding of our fam molecule, our uncleaved reporter molecules are captured onto the first line. Now, when this reporter molecule is cleaved by our CRISPR-Cas system upon activation by interaction with our target ITS region, cleaved reporter molecules travel further up the strip, which is eventually captured on the second line, indicating a positive result.
These results reported by the CRISPR-Cas reaction are captured through our sensing methods.
To start the process, we started out with mapping a plethora of options, and we narrowed these options down based on our constraints and requirements. Now, for fluorescence for excitation, we're looking at laser or LED, and we're testing both to find which one is the best. For detection, we're looking at a photodiode module. And to eliminate the need for an expensive filter, we're looking at statistical techniques such as one-tail t-tests to see and compare its performance with conventional filters, so see if we can get rid of this. And for LFA, we're using smartphones to take images and then using the intensity of coloration on the test line. We're combining this with machine learning methods. We're trying to get quantitative results. And for these options, we have already designed the experiments and we're currently in the process of testing them.
Data collection is crucial in disease management.
Rapid analysis and distribution of data is important for infectious disease management. That is why we're developing a comprehensive database that can be used to retrieve and manage diagnostic data, and an API that can integrate to any existing surveillance system. Mapping users and understanding the why behind our device necessitated an understanding of where diagnosis falls in infectious disease control. We divided our user mapping process into stages depending on the disease dynamics, which let us identify stakeholders that we later reached out to, ranging from field researchers to mycologists, that provided invaluable insight and understanding the problem at first and then crafting the solution. Even in the current stage of our problem, we're prototyping the device components. We've benefited from expert opinion that continues to guide us. We've documented nearly every detail of our research and overall project, and we've made it available to the iGEM community via our docs. We've published pages of case studies, experiment planning, stakeholder input, essentially everything that has guided us so far in developing the diagnostic product. Through this rigorous approach to research and design, we're hoping to deliver an effective solution to animal infectious disease control.
Beyond the project, the NYUAD iGEM group has always felt the responsibility of raising awareness on science-related topics and the uptake of technologies since its inception in 2016. This year, we began EdBio, an initiative targeted at synthetic biology education in secondary schools. We are working to develop a toolkit with physical and AR components for schools that demonstrate basic concepts like DNA replication or RNA translation. The EdBio team has taken part in a 3-month-long entrepreneurship program at StartAD, a global startup accelerator, and has received a prototyping grant of amounting to $2,500. We also engaged with the public at the annual Abu Dhabi Science Festival, the largest science festival in the region, as science communicators, where we designed synthetic biology experiments and played the role of science communicators. Follow our journey by checking out these links.